right, guys, this is the recording for Traits and Reproduction 2.3, 2.4, kind of squished them together. Remember that the second lesson of this week is going to be a hands-on lab. The supply list was sent to you as an announcement on Monday, so if you would like to participate, it is highly recommended, and most of those supplies can be found around your house, like soap, dish soap, salt, things like that. It'll be fun. We're going to do some DNA extraction on strawberries and our own cheek cells. So should be good. Highly recommend you join me doing that. Um, the assignment for today is here. Attendance, hopefully you took it on the day you needed to. So the assignment today has you looking at a simulation to start. And you can see it's hyperlinked here. And you are going to pick a feature. Pick any feature, but do not pick the body size. There are a bunch of other options, and it wants you to pick one. So example being, if I did let's do venom. And it wants you to pick one spider that has two of the proteins that are the same. And then it wants you to pick one spider that has two different ones. A kind of hint is our gene versions here. So Zora has no venom, and I can see the gene versions are V3 and V3, and then there is protein 3 in here only. So there's only one type of protein. So going back to my chart, Zora would be a good spider to put for only one type of protein here for this feature of venom. Then it wants to find a spider that has two different versions. So I'm going to see here, Otis has V1 and V2, and it has protein 1 and 2 in here. There's 2, there's 1. And so Otis would be a good protein or a good spider that has two different proteins. The trick of this is down here, I wanted you to tell me what you could do to cause the spiders to have the same proteins. Either you want to make one spider have two of the same like the other one, or vice versa. So example, if I want Otis to have two of the same, I could change the gene versions up here and notice how those proteins will change. And it does not change its trait. And then you could change it to other ones and see. So that is one way. The other way that you could do that is you could pull the proteins from down here and pull them in to see what happens. So those are those two versions. And then sometimes it might affect the spider's trait, sometimes it might not. You need to tell me based on what you did with your feature and which spiders you chose. The other thing down here is this picture that we have seen a bunch of times. We have our 4 offspring and they have a different variety of silk flexibility. And so you can see inside their cells, they have the different proteins, and you also can see they have gene versions here. And we have our options. And so what I was showing people, and I will write it on here, hopefully if it lets me. So F1 is going to be the protein that has 4 prongs. And this is all information you can get from the simulation. And then F2 is the protein that has 1. And then F3 is the just straight line. So you're going to look into the cell and so you can see, oh, I only see F1 and F1. So that would make this one homozygous, which we have our 2 terms, heterozygous and homozygous. Homozygous is when the 2 gene versions are the same, and heterozygous is when the 2 gene versions are different. And so you're going to go through each one. So going down here, you're going to see there is a box that you're going to fill out, and I started filling it out for you. You're going to tell me what gene versions they're going to have and whether it's homozygous or heterozygous. Your chart might not have this second column here. You can quickly add that by just right-clicking, and then it will pop up, and you can say insert column right. And so you can tell me heterozygous and homozygous. Remember, homozygous is when they are the same. So I had two F1s, and then heterozygous is two different, which is why I had an F1 and an F2. Then down here, we have some more information about our ACTN3 protein, which, if you remember, was our sprinter's gene and just our overall running ability trait. So we notice now we have some new information that the two gene versions that affect running ability are A1 and A2. But A1 is the one that instructs to create that ACTN3 protein. So that means that if it has two A1s, it's probably going to be someone who runs very well. So you're going to see down here there are three pieces of evidence. You're going to read those three pieces of evidence, and based on what you find in those three pieces of evidence, you're going to answer this question. So the first one I did for everyone in class to give an example. So the evidence was that for the running ability feature, runner 1 only had ACTN3 proteins in her cells. And since I read up here that my A1 is the one that instructs for that protein, I figured out that that would mean that runner 1 would need to have two A1 gene versions, and that would mean that it was homozygous because it's two of the same. And then that would also mean that this runner would most likely be a fast runner or have a good running ability because of that fact that it has two of the proteins that instruct for the, or two of the gene versions that instruct for that protein. And so you'll do these two below on your own. And then you want to make sure that you turn it in, and don't forget about our lab on Wednesday or Thursday, depending on when you have class. I hope you can gather those materials and join me in doing it. It's a good one.